All right, good afternoon, everybody. We are here for the second to last AP Biology review. We're talking about heredity and gene expression today. Give me a second to get all set up. We've got students in the class, so you'll hear them in the background. They might shout out some questions. All right, I just want to remind you what gene expression, heredity and gene expression refers to. Heredity is the unit that's on Mendel. We learned Punnett Squares during that unit, and we talked about meiosis. And then gene expression was about DNA and RNA. We talked about the trucks and the ribosomes and building proteins and that kind of thing. I'll try to go fast. Those of you that are watching it online, you can speed it up or slow it down as needed. So last time we talked a little bit about mitosis. I just want to compare and contrast mitosis and meiosis really quickly. Remember, mitosis, you get two cells. I always emphasize the T. Mitosis is two cells, and it's just cloning. So that's when you skin your knee and you need to replace the skin cells. The skin cells just divide. They've copied the DNA. You started with 46 chromosomes. You end with 46 chromosomes. Meiosis, the only place in our body that meiosis takes place is in the sex. What is the word I am looking for? So the testes and the ovaries. I don't know what I'm trying to say there. Okay, so that's the only place that meiosis is going to happen. And we're going to produce, if it's in a human being, we're going to produce sperm or egg. If it's happening in a plant, we can produce pollen. Plants also have eggs. So the flowers have both pollen and eggs. So that's where meiosis is occurring. Meiosis is going to produce 4 cells at the end, and they have half of the DNA. And why did I say it that way? Because they are haploid. So just to remind you, so when 2 haploid cells come together, they form a diploid organism. And all of our cells in our body are diploid except for our sex cells, which are the haploid cells. All right. And then just reminding you about chromosomes in a diploid cell. Diploid cells have 2 copies of each chromosome. What you're looking at is called a karyotype, and that's a photograph of somebody's chromosomes. And it looks like they're X's, but they're not. That's just one is from mom and one is from dad. If we could really close up on them, and they do it in the little diagram here, this right here looks like an X. That's one. And then this is a different X right there. They just both happen to be bending and looking like an X. Okay, so the pairs, you get one from mom and one from dad, and they're called homologous. And the reason it's got the prefix homo on it is that it means that they have similar DNA. So homologous pairs might have, they both have the gene for eye color, for example, but moms might say a gene for blue eyes and dads might say a gene for brown eyes. So they're not identical, but they carry similar DNA. And then don't confuse homologous pairs with sister chromatids. Sister chromatids are the 2 sides of an identical chromosome, and they separate when the cell gets ready to divide. So during anaphase apart, they get pulled apart, and then the 2 copies go to opposite sides of the cell. All right, moving on. And then just again, chromosomes in a diploid cell here. The stage right here is prophase. They're scattered all around. Remember, when we're talking about mitosis, it goes prophase, metaphase, anaphase, telophase, PMAT. So prophase, they're scattered around. Metaphase, they're in the middle. Anaphase, they pull apart. And then telophase, 2 cells. You've divided in 2. So that's for mitosis. But now we're looking at meiosis. So instead of IPMAT, we have IPMAT PMAT. It divides 2 times. And on the screen, give me just a second. On my video, I was covering it up. So you can see there's, for the meiosis, there's a cell division here. And now we've got 2 sets of cells. And then they divide again for a total of 4 cells down at the bottom. The first half of meiosis is denoted with a Roman numeral 1. And remember that meiosis, the whole purpose of meiosis is creating new genetic combinations and adding genetic diversity to a population. So we're not creating clones of ourselves. We're trying to make sperm and egg or pollen so that we can genetically recombine and have diversity. So so in addition to the diversity that comes with a unique sperm and a unique egg coming together, they also do crossing over. And I'll show you that in just a second. The chromosomes cross over. All right, so the chromosomes line up side by side, not single file. And this is where the chromosome number then gets cut in half. The second half of meiosis is denoted with a Roman numeral 2. So it might be like P1 versus P2. And that's how you know that that's the second half of meiosis. And the second half of meiosis is identical to mitosis. It's just that it has half the number of chromosomes. So the second half of meiosis would have 23 chromosomes. Why? Does anybody remember why it divides again? Already right here, it has 23 chromosomes. Why do we divide a second time? Because they're still bivalent. The chromosomes are still bivalent, and so that's an extra copy of the DNA. So it's going to divide again so that all the chromosomes are monovalent. That's it. So this is the crossing over that I was telling you about. This shows two bivalent chromosomes that have sat next to each other, and now they're crossing over. I often showed it like with my fingers crossing over, and they're actually trading pieces of DNA. So it says the homologous pairs connect to each other. That's called synapsis, forming structures called tetrads. The prefix tetra means 4. At each connection, it's called a chiasma. DNA is exchanged and swapped. So you are creating within your egg or sperm unique chromosomes that have never existed on this planet before. They are a unique combination of your biological mother and your biological father's chromosomes. They're melding together. And now that's what you're going to pass on to your offspring is a chromosome that neither of your parents had. It's a melding of the chromosomes that your parents had. And that's increasing genetic diversity. All right, what are the 4 things at the end, just to make sure you understand what's happening? Meiosis? Give me words. Gametes. What else? Haploid. They're haploid cells, and if we're going to be really specific, they are blank, blank, and blank. You know it. They are sperm, egg, and pollen. So that's what you're looking at the end. Even though that doesn't look like 4 sperm, we would draw 4 little tails on it, and that would be 4 sperm at the end. All right, and then what is genetic recombination? We're talking about recombining our DNA so that we're not clones. And so we're adding to genetic diversity. And there's several things that can increase genetic diversity. Number 1 that I mentioned here is crossing over. That's what you're seeing in the diagram there. That's increasing genetic diversity. Mutations. That's when we're copying the DNA during replication, which we'll talk about today. They make some mistakes, not very many, but a few mistakes slip through. That's often what leads to cancer, but it can also lead to new phenotypes. So that means new physical, they have a different eye color than anybody else, or they have a different, ability to handle this poison than anybody else. And that's due to mutations. And then another thing that increases genetic diversity, so we call this genetic recombination, is the idea that a unique sperm and a unique egg come together. And so really the odds of ever having 2 human beings that are identical is 0. I mean, you could do the math on it and say, well, KJ, it's not 0, but nowhere near that many human beings have ever existed on this planet. So nobody's going to be the same based on that. All right, so now we need to talk about Mendel. You don't need to really know much about Mendel, but he did come up with 2 laws. Remember that he could not see the DNA. He didn't know what the DNA was. His 2 laws are based on his experiments in the garden. He's looking at traits. He crosses different types of plants, and then he looks at how they turn out. His first law is called the law of segregation. And you guys know the word segregate means to separate. So we see segregation in the United States in the early half of the 1900s. We see segregation. So they're separating. So the gene that came from mom and the gene that came from dad, they separate. That's his law of segregation. And then his law of independent assortment. This picture is about the independent assortment, but I feel like the picture is super confusing. The basic idea is that you have 20 to 25,000 genes that are on 46 chromosomes. It's not that all of moms line up on the left and all of moms line up on the right. They assort independently. So you might get 20 that are moms on the left, and you might get 26 of dads on the right. It doesn't matter. So it's just they're assorting independently. It's not always moms on the left and always moms on the right. All right, moving on. Let's just talk Punnett squares really quickly. I'm going to assume that you remember how to do a Punnett square. So just remember, just a quick review. Remember that on the outside of the Punnett square is the parent's gametes, so the sperm or the egg. And then on the inside of the Punnett square are the possible babies when that sperm and egg come together. And so what I wanted to remind you of is how to handle a really big Punnett square. So you can handle, you guys are good, everybody's good at doing the small Punnett square where it's just like one trait, big T versus big T little t, and you fill in the Punnett square. But what if you have many, many different traits? So in this example, I gave you a dihybrid example. You could do a really big Punnett square, or you could do one Punnett square for each trait and then multiply the results. And we agreed that this was a lot easier. I'm going to walk over to the board here really quickly and just remind you how you did that. So you did one Punnett square for the yellow trait. Look at it down here. Here's the yellow trait. And you did one Punnett square for the round trait, whether it was round or wrinkled. You did one Punnett square for that one. And then you multiplied the results. So the question was, what are the odds of having a yellow round pea? These were for pea plants. Then you multiply, okay, this is yellow, this is yellow, this is yellow, this is green, right? Yeah, this is green. So it would be 3/4 yellow, times how many are round? Round, and this one is wrinkled, so 3/4 wrinkled. So 3/4 times 3/4 gives you the odds of having a yellow round pea plant. Do you feel like you remember that? So we were able to, on a test, put together, I think I gave you a problem that had 4 or 5, and you made one little square, and then you just multiplied the odds to tell me what are the odds that the offspring would have this trait, this trait, this trait, and this trait. Are you looking at me blankly? How do you feel about it? Are you okay? Oh, yeah, it's familiar. Good. Okay. Just wanted to remind you of just some vocabulary words. A test cross is what Mendel invented. It was the way of figuring out whether an organism is homozygous or heterozygous. So it says a method for determining the genotype of an organism that expresses the dominant phenotype. So, for example, if you see a purple flower, it could be big P, or it could be big P, little p. How do you test cross? How do you find out if it's homozygous dominant or heterozygous? And the test cross says you made it with a recessive. Did that make sense? Made it with a recessive. If you made it with a homozygous dominant, then they're all going to be purple and you won't have found anything out. But if you made it with a white flower and some of them are white babies, then you know it's a heterozygous parent. You know that one was originally heterozygous. So that's what we call the test cross. All right, moving on. Codominance versus incomplete dominance. Codominance, they both show up. The classic example is blood types. If you get type A from one parent and type B from another parent, then you have type AB. You show both of those proteins on the surface of your red blood cells. Remember, there's also type O, which is recessive. That has nothing to do with codominance, but just telling you that there's type O blood and it's recessive. Okay, as opposed to codominance, as opposed to incomplete dominance, they blend. So this is where, in this example, there's a red allele and there's a white allele, and they blend to make a pink baby. If you have two reds, it's red. If you have a red and a white, it's pink. And if you have two whites, it's white. So where did this pink come from? Because there's no pink gene. It came from the blending of a red and a white together. So that's incomplete dominance. It does say in the learning targets that you need to be able to solve Punnett squares using codominance, or if they have codominance or incomplete dominance. All right, and then just a quick reminder too. I say quick every time, don't I? I'm trying to go fast. That's why I keep saying quick. Polygenic, the prefix poly means many, and gen means genes. So these are traits, which is true of almost everything in your body. It's controlled by more than one gene. Does anybody remember? I think that was in this class. How many genes teeth take? Was that in this class? Did we talk about that? They think there are 300 genes involved in determining your teeth. So that's an example of a polygenic trait, which is shocking. I had no idea. Skin color, I've heard anywhere from 6 to 12 genes determine our skin color. So we don't have dark and light. We don't have lots of melanin and no melanin. We've got all these shades of brown in between, and that's because one gene had melanin and the other one didn't. But this one had no melanin at all. But then this one had 2 genes for melanin. And you add that all up, and that makes the more melanin we have, the browner our skin gets. Other examples would be eye color. We don't just have brown or blue eyes. And height. We're not just tall or short. We've got all these variations in between, and that's because it's controlled by lots of genes. All right, reminding you about sex linkage and how to do a sex-linked Punnett square. Sex linkage, 99% of the time, means that it's carried on the X chromosome. Sometimes it's carried on the Y chromosome. So let's just talk about that really quickly in case they give you a trick. If it's carried on the Y chromosome, who's going to be affected by it? Males. If it's carried on the X chromosome, who's more affected by it? Wrong. Still the males. Okay, who just said it? What'd you say, Abby? Because they don't have an extra X chromosome. Okay, they don't have this backup X copy. So if a guy gets any kind of flaw, I don't know the right word for it, something in the genes, and he does not have a second copy, then they automatically show whatever that gene is. Whereas females have 2 copies of the X, one of those X copies goes away, and they just have this dominant X copy. And so that is sort of a backup copy is the way I've been teaching it. So would you please look at how to do a Punnett square when it's sex-linked? If it's on the X chromosome, which usually that's how it will be shown, if it's on the X chromosome, you put a capital N for dominant and a lowercase n for recessive only by the X chromosome. You have to show who's the male and who's the female. You don't do that with other kinds of Punnett squares. So other kinds of Punnett squares, because males and females are the same otherwise, it would just be like big N versus big N little n. But when it's only carried on the X chromosome, then it's going to be X big N Y, no N by the Y, versus X big N, X little n for the female. And then your offspring have to show the X's and Y's. Any questions about sex linkage, meaning it's attached to one of the sex chromosomes? Okay, shout out if I'm going too fast. So in general, gene linkage refers to genes being on a specific chromosome. And when we say gene linkage, we're talking about 2 genes that are on the same chromosome. So if you inherit in this diagram, if you inherit gene capital A, you also inherit gene capital B and gene capital C because they're all on the same chromosome. Now look at the diagram. What is the only way that you would not inherit gene capital A, capital B, and capital C at the same time? What's the only way that a person wouldn't inherit all 3 of those at the same time? If crossing over has happened and separated those genes. So that, just trying to remind you, is how we determine determine the maps, the chromosome maps. How often they get separated tells us how far apart they are. So if they get separated a lot, then they must be very far apart on the chromosome. But if they almost never get separated and they're always being inherited together, they must be really close together on the chromosome, and so they're not crossing over very often. All right, so try this puzzle. It says if the genes are on different chromosomes and inherited separately, we would predict 2 Punnett squares, and we multiply the probabilities together, and that's what we would predict if these genes were carried on different chromosomes. However, and it was a 1-to-1 to 1-to-1 ratio. I will say that this was the least favorite thing for most of you, was dealing with these ratio things. So I'm going to remind you of it, and then we're moving on because I don't think you need to obsess over it. Okay. So when the guy who did this, Thomas Hunt Morgan, when he crossed these fruit flies, it said predicted ratios if they were on different genes or different chromosomes. It should have a 1-to-1 to 1-to-1 ratio. If they were located on the same chromosome, we should see a 1-to-1 to 0-to-0 ratio. I'm looking right here. That's his prediction. So then he really crossed these fruit flies with these traits, and he got craziness. He got kind of his 1-to-1 ratio, but not quite. He's supposed to have 0 of 2 of them, and he's got 206 and 185. Can anybody tell me what happened? What is this phenomenon? We already said it just now. We just said it. What caused them to not be inherited correctly? Did somebody say it? Did you say it? What'd you say? Crossing over. Yeah. So crossing over separated these genes. So he was predicting that it would either be 1-to-1 to 1-to-1 or 1-to-1, 0-to-0. And he got a little bit of a mixture, and that's because sometimes those chromosomes separated, those genes separated from each other on the chromosome, and that's why you don't have a perfect ratio on that. Okay, that is all we're going to talk about that one. Chromosome maps, I already said this. The frequency that they recombine or cross over, the frequency that they cross over tells us how far apart they are. So on this one, between B and VG, they cross over 17% of the time, whereas the CN and VG only do 9.5%, and the B and CN are only 9%. By the way, they never add up perfectly, because there's some double crossing over. I don't know if you remember that, but they cross over and then they cross over again. So these numbers never match this. That's okay. But we can see that these two must be closer together on the chromosome because they're only 9% apart. And these ones must be further apart on the chromosome because they're 17%. I gave you a test question where I told you the the percent of times that they recombine, and then you had to put them on a chromosome, and you guys did really, really amazing on that one. All right, reminding you of pedigrees. Males are often shown as boxes and circles are females. What's interesting to me, I don't know as our society changes and we start looking at gender and that kind of thing, whether they'll change these. Part of the issue that I think they don't change them is that this is entirely based on sex chromosomes, not based on gender. Would be how a person feels about themselves and not necessarily what their sex chromosomes are. So that's why there's still pretty clear box and circle. But I'm guessing that with time, we're going to start seeing pedigrees change a little bit to reflect also gender. But right now they don't do that at all. Okay, so let's take a look. It says, I didn't switch it. Is the red genetic trait due to a dominant or recessive allele, or are both possible? So in this case, the ones that are red, they are affected by whatever the trait is. You need to be able to look at it and say, hey, that looks like a dominant trait or a sex-linked trait or a recessive trait. Can you figure it out? I actually don't know the answer myself, so I'm going to have to look at it with you. Let's actually do it on the board so that we don't have to wait for people to solve it. So just giving you some ideas. So if this is due to a recessive trait, then everybody who's colored in red is little n, if this is due to a recessive trait. This individual is big N something. We don't know what their second letter is. I can tell because he's got a son, if this is correct, because he's got a son that has two little n's, he got one of the little n's from here. The second little n must have come from this dad. So far that works. Could they have a normal son? Yeah, they could. So far that's working great. Do you see anything on here that would not work for it to be recessive? This one got it from here. So this one must be big N, little n, in order to have little n's. Give me just a second to see if there's any that are not possible. Do you see any that couldn't be? Anybody agree? I think it's a recessive. It could be a recessive. Let's try and see if it could be a dominant trait. So now I'm going to switch it up. And everybody who's red has one copy. They could have a second, but they have one copy of the capital N. So we know this individual is big N something. That's why they have it. This individual is little n. That's why they don't have it. So this individual is little n. Is that possible? It sure is, because this person's second letter could be a little n. That would explain it. Is it possible that these have it? It sure is. Trying to think if there's anything, if the dominant parent passes it on. One of the secrets to, so I'm going to guess that this is all possible. One of the secrets to the difference between, so dominant and recessive are both possible for this one. In a recessive, it's possible for a child to have it and the parents don't. In a dominant, that's not possible. So if a child has it, they have to have a parent that has it. That's the only way that would happen. Okay, I'm going to move on. I think I give you one more of these as a little thing that you could try. Autosomal dominant, that means your regular chromosomes, not the sex chromosomes. So it could be autosomal dominant, autosomal recessive, sex-linked recessive, and sex-linked dominant. I don't even remember off the top of my head. So do we see any kids that have it and their parents don't? Yes. We do. Down in the bottom, number 9 has it and their parents don't. So that means this is not autosomal dominant for sure. Let's see if it could be sex-linked dominant. Nope, it could not be sex-linked dominant either. So we know it's not those. So now, could it be sex-linked recessive? What would you expect to see typically if it's sex-linked recessive? More boys than girls. So girls would be quite rare. That doesn't mean it's not possible for the girls, but the girls have to get it both from their dad and from their mom. So let's take a look. This could for sure be a recessive, an autosomal recessive. Let's just see if it could be a sex-linked recessive also. Is there any way that it might not be? Let's take a look. Give me just a second to eyeball through it. So let's look over here. Could this boy have it if it's sex-linked and his parents don't have it? Is it possible? No. It is. It is possible. He would be X, if it's recessive, he would be X little n Y. He got the Y from his dad, and he got the X little n from his mom. And his mom must be X little n and X again. So this could be either sex-linked recessive or autosomal recessive. They would both be correct. All right. Moving on. Reminding you that your nucleus is not the only place that has DNA. And a lot of you are getting this, you're hearing about this from Ancestry.com and that kind of thing, where you can send in your DNA and find out about your ancestors. So you have mitochondrial DNA. And in plants, there's also DNA in the chloroplasts. So that's just that's called non-nuclear inheritance. Nondisjunction, that's when the chromosomes are supposed to line up side by side, and they do, but then they're supposed to separate. And in a human being, you should have 23 that go to the left and 23 that go to the right. In nondisjunction, that's an error where two of them stay stuck together. And so you end up with, if these were the, let's say these are the sperm at the end, these two sperm end up with extra chromosomes, and these two end up missing chromosomes, one chromosome. This is an extra one chromosome, and this is missing one chromosome. Or vice versa. There's several different possible combinations. Who remembers probably the most famous genetic disorder that's caused by nondisjunction? Good. So nondisjunction leads usually to an egg that has an extra 21st chromosome, and then a normal sperm fertilizes it, and we end up with a baby that has Down syndrome. The two other that are listed, the two other nondisjunction, or they're called trisomies, because they have three of a particular chromosome. The other two that are on your learning targets are Klinefelters, which are boys that have inherited an extra X chromosome. So remember, they're the ones when they hit puberty, their voices don't lower, they develop breasts. They're still boys in every, men in every sense of the fashion, but they have too much estrogen. And so that causes some of the female characteristics and hides some of the traditional male characteristics of going through puberty. And then Turner syndrome is the opposite. Turner syndrome is girls that are missing an X chromosome. So they're perfectly healthy, normal girls. They tend to be short. They tend to be infertile. And occasionally they have learning disabilities. You don't need to know that, but just telling you that that's another example. In this case, one of either the egg or the sperm was missing a chromosome. And so these kids have 45 chromosomes. Down syndrome and Klinefelters have 47 chromosomes. They have an extra chromosome. All right, and then just reminding you that there are some organisms that are polyploid that have more than, we are diploid organisms. There are some organisms for whom it's normal to have more than two of every chromosome. If only that were true for us, that would be good. It creates what they call, this was the word in your learning targets, they call it hybrid vigor. I'm going to try to highlight it. I can't. Oh well. Hybrid vigor, which just means, just like how girls have an extra copy and it helps them with some of the sex-linked genetic traits, polyploids have an extra chromosome. And so if something's wrong with one of them, they have an extra chromosome they can use as a backup. All right, that's it. We're going to talk, that's not it, sorry, I'm going as fast as I can. That's it for Mendel's unit and for talking about heredity. And now we're going to just talk about DNA. This is a little bit faster, I think, I hope. Still aiming to get you out of here by, I tried for 3:30. You do what you need to do. Just reminding you really quickly about your DNA structure. We already talked about this in biochemistry, so I'm not going to say a whole lot about it. Remember that the two sides of DNA are joined by hydrogen bonds, so they separate really easily. The strong covalent bonds that run along the backbone, those are called phosphodiester bonds. I don't think you would ever need to think of that word. But if you saw that word, you would go, oh yeah, that's that covalent bond in DNA. But I don't think you ever have to come up with it. It does say in your learning targets, it mentions knowing the difference between the purines and the pyrimidines. These are the nitrogen bases. And the way that I always remember it, the word pyrimidine has a Y in it. And so does thymine and cytosine, and those are the pyrimidines. And then uracil replaces thymine. So these guys have one ring, and the purines have two rings. And if I just show you really quickly, I'm going to go back to the previous slide. In DNA, you always have a single ring across from a double ring, and a double ring across from a single ring. So there's always a purine across from a pyrimidine. And if there's ever not, it creates a misshape in the DNA molecule. Okay, DNA replication. Remember replication? Okay, let's go through it. So replication happens in the 5' to 3' direction. Do you need to say that? No, but it is absolutely possible that they would show you a diagram and they would say, what's wrong with this diagram? And you would be like, oh, it's supposed to be 5' to 3', and they showed it as 3' to 5'. So that is something that I could see them testing you on. Does anybody remember what the term semi-conservative meant? Listen to you guys. Good. Shout it out to yourselves. Even if I don't hear you exactly saying it, helps. So for the people who are online, it means that you take the original old strand and then you copy new strand onto it. So there's an old and a new on every strand of every strand of DNA. All right. So the helicase is the enzyme that unzips it. I don't think helicase is shown on this particular diagram. Topoisomerase untwists it. So you'll probably see a diagram that has that on it. Nothing that you need to come up with on your own, but you'll probably see that. And then RNA primers. It does show, I think, an RNA primer somewhere on here. Yep, it's showing primase. RNA primers basically tell the DNA polymerase where to begin. That's all not the hardest part. The hardest part was the Okazaki fragments. So remember, the leading strand happens continuously. If DNA polymerase reads an A, it attaches a T. If it reads a C, it attaches a G. Very straight and fast. But on the opposite strand, it's going in the opposite direction. And so the DNA polymerase makes these Okazaki fragments, and then they have to be joined by the enzyme that's called ligase. Its job is just to join them so they don't fall apart. All right. Replication, remember, occurs during the S phase of the cell cycle. This is the number one time where we get mutations. So when the DNA is being copied, sometimes our DNA polymerase makes a mistake. We have enzymes that proofread it, but sometimes they get screwed up. So after proofreading, the error rate is about 1 in 10 billion. You have 3 billion A's, T's, C's, and G's. I had to think that number through. I think that's right. And then mutations accrue over time, leading to higher rates of cancer in older adults. All right, so now we're going to talk about transcription. I hated this word because I feel like it doesn't mean anything to anybody. So transcription is when we look at your DNA and we make a copy one side, and that's the RNA message that leaves the nucleus and goes to the ribosome and tells the ribosome what protein the DNA wanted it to build. That's transcription, is making the RNA. Translation is actually building the protein. I wish we didn't use those terms. It's not really what I want. So on here, it just a couple reminders. Instead, this time the enzyme is called RNA polymerase instead of DNA polymerase, and it only copies one side.
The way I remember, like, transcription and translation.
Let's hear it.
It's like translation. It sounds like it's later.
Oh my gosh, I love silly tricks like that. Rachel said the way that she remembers translation is that translation happens later. I love it. Transcription first, translation later. Perfect. That's super helpful. All right. And then just reminding you of a few things. Notice the Tata box up at the top. That tells the RNA polymerase that that's a beginning point. Notice the transcription factors. Should I give you guys a break? Is it too much? I want to go fast for you, but I don't know. I'm going as fast as I can. Okay. Trying to go as fast as I can. All right, here we go. And then remember that the side that it's copied is called the template. And the side that is not copied is called the non-template. So the non-template or coding strand. So not being copied. I don't know if that helps, but it's called the coding strand. All right, and then what's the new nucleotide that we We had A, T, C, G in DNA. What's the new one? U instead of T in the RNA. Good. Okay, and then remember eukaryotes, we have really complicated DNA that has introns and exons. And so all kinds of things have to be done to our mRNA before it can leave the nucleus. We add a poly A tail, we add a 5' cap, and then we splice it. And I'm showing you what happens with splicing down below. So alternative splicing, so the introns are removed and the exons are put together, and they can be put together in different ways. And notice, the diagram down below is showing 3 slightly different proteins, and that's because the exons were put together in different ways. I think we learned in this unit that there was a Drosophila, a fruit fly gene that could be put together in 19,000 different ways. So it adds a lot of variation. Okay, this is what a ribosome, this is one image of a ribosome. Remember, ribosomes have a small subunit and a large subunit, and they clamp around the mRNA, and then the trucks pull in and drop off their amino acids. So here's an example of a truck. They're drawn in many different ways. The thing I want you to see when you look at these trucks, the tRNAs, this is where the amino acid attaches here. And down here is called the anticodon. It's the opposite 3 letters. Here again is where the amino acid, it's a different drawing, the same idea. But down at the bottom is the anticodon, the opposite 3 letters. And then this is how your book drew it. The amino acid attaches here, and then there were the 3 letters. There's so many different ways of drawing a tRNA. Look for the amino acid attachment site in the anticodon on the bottom of it, and that will help you know that you're looking at the trucks. Okay, so the genetic code, what's important is that it is universal across all life, and that is super interesting because now we can do these crazy genetic engineering experiments where we can take a human gene and put it into a bacteria, a human gene for insulin or a human gene for human growth hormone. And we can put it into a bacteria, and a bacteria will build it because it is universal. They all use the same 3 letters to denote the different amino acids. All right, and then remember there are start and stop codons. Every mRNA starts with the AUG start codon. Wobble refers to the fact that you could have a mutation. Look at this CGU one here. If it was supposed to be CGU, but we had a mutation and it changed to CGC, we would still get arginine as our amino acid, and so that's good. So that protects us against too many mutations affecting us too terribly. You are very, very welcome. Okay, so this is just the overview. Here's transcription that's happening in the nucleus. Here's the mRNA. We've added the cap and the tail. We've cut out the introns. We've put the exons together. Now it comes to the ribosome. Now the trucks pull up with their opposite anticodon, and they drop off their amino acids. They all bond together, and then when they hit a stop codon, the polypeptide is released, and then it can be packaged or whatever needs to be done. All right. Prokaryotes don't have introns and exons, so they don't do anywhere near the complicated stuff that we do. So as fast as the RNA polymerase is making an mRNA, their ribosomes clamp on and start making the protein. So they are much, 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 much faster organisms than we are. Their biological processes are simpler and they're streamlined. We are very, very slow. We're glacial in comparison. Did you know glacial means slow? I like that word. Like a glacier, slow like a glacier. All right. Retroviruses. The thing that makes a retrovirus retro, HIV is the example, is that instead of having DNA in it, it has RNA. So it goes from RNA to DNA back to RNA. and then to the protein. And the problem is that it has an enzyme called reverse transcriptase that makes bajillions of mutations. And so everybody who has HIV has their own variety of HIV. All right, and this was probably the hardest set of notes on the gene regulation unit. Gene regulation refers to turning genes on and off. Remember, you have 25,000. You don't want them all activated at the same time. We want to save energy, and I don't want a big toe growing out of my cheek. So the gene for growing big toes has to be shut off. That's gene regulation. We studied two different types of genes in bacteria called operons. There was the lac operon and the trip operon. The lac operon is an example of negative gene regulation, and that's because the repressor proteins shut them down. So if there's a repressor that turns it off, we say that's negative. I guarantee you they're going to ask you something about negative and positive feedback. So if it shuts it down, it's negative feedback. If it ramps it up, it's positive feedback. So we saw the lac operon shut it down. This is the example of the lac operon, and it shut it down. But then we also saw, give me a second, we also saw this thing called CAP. And it was like turbocharging the lac operon and made it happen a lot faster. And so that's an example of positive regulation. So that was the opposite of negative regulation in that case. All right, and then your learning targets specifically mention small mRNAs. It's surprising to me because I don't think they're all that important, but I must not know enough. There's probably a good reason that we are really interested in small RNAs right now. So the general idea, small RNAs can shut things down. They can turn off genes. So it says they degrade target mRNA molecules so that doesn't turn into a protein. They cause it to break down. Or they get in the way and they interfere. And so then the ribosome can't read the mRNA. But either way, the protein doesn't get produced. So small RNAs can shut everything down. Remember, there's two main types of mutations. Point mutation is where we just kind of change one, we substitute one letter for another letter. And that usually isn't too bad if it's just one letter. But a frame shift mutation is where we delete or insert. And that screws up the groupings of 3, the codons, for everything after that. And so everything is ruined in that case. All right, and then we're almost to the end, you guys. How do prokaryotes evolve? Remember that prokaryotes clone themselves. So there were 4 ways that we learned. They can suck up DNA from the environment and get new DNA. That's called transformation. They can get DNA from each other through a process that's kind of like bacteria sex, but there's no such thing as a male or a female. And so they're just getting DNA from another bacteria. That's called conjugation. Transduction is when they get DNA from a virus. A virus accidentally picks up some DNA from one bacteria and transfers it to another bacteria. And then the last way is called transposition. It's called transposition, and that's where they sometimes call these jumping genes, where an antibiotic-resistant gene, for example, that's here, can now move over here to the smaller plasmid. And then that's a way that they can then be transferred. Antibiotic resistance can be transferred from one bacteria to another, is if it's jumped into a plasmid and then the plasmid gets transferred through conjugation or through transformation or some other way.
What was the first one?
The first one was transformation. This is called transposition. The first one was transformation.
What's that?
Picking up the DNA from the environment. Okay. So if a bacteria died and its guts are spewed out, it can pull it up. Okay, just about done. Want to remind you about restriction enzymes. They cut the DNA. They look for patterns in the DNA and they make cuts. Sticky ends are super useful. Sticky ends are super useful for genetic engineering experiments. We cut a gene with the same restriction enzyme as we cut the bacteria's DNA, and then they stick together. And so I can insert the gene for human insulin into a bacteria if I've cut it with a restriction enzyme that has sticky ends. We talked about PCR. I think all you need to know about PCR is that you make copies of the DNA. It's possible they would ask you for the steps of PCR, not that you would have to say them, but like they might make a multiple choice question out of it or something like that. But basically, it's a machine that can take this tiny drop of blood, you put it into a test tube, you turn on the machine, and a few hours later, you have billions of copies of that DNA. And now you can do next. Oops, sorry. These are the steps of PCR. And then you can do electrophoresis, and you can do a DNA fingerprint of that DNA that you just made a million copies of. So remember with DNA fingerprinting, a few years ago, there was a question that asked you to make a DNA fingerprint on the test itself. Remember that all the lines tell you is how big the fragment of DNA is. They're negatively charged. They go toward the positive end. The small fragments travel faster. The large fragments don't travel as fast. So if it's close to the bottom, close to the wells, it's a big fragment. If it's near the top, it's a small fragment. And you guys, 1 second more, and then we're done. La, 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 la. Sorry. Moving one more thing. La, 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 la. DNA sequencing just refers to, we did the Human Genome Project where we figured out all of the A's, T's, C's, and G's in the human genome, and we've done that for many species. We don't know what all the A's, T's, C's, and G's mean, but we have figured out all the letters, and now we're trying to figure out what they mean. Whew, all done, everybody. You survived yet another AP biology review. You did great. You guys are amazing.